Yes, it does. You good?
Yes.
Great. Thanks.
So, good morning, everyone. So, today I will describe to you the South African pear stony pit disease experience. So, this was a project conducted by myself, Professor Hanno Maria, and our MSc student, Kaylee Bogart. So, what is pear stony pit? Pear stony pit is when fruit growth is restricted, resulting in dumpling, pitting, and deformed fruit. These deformed fruit can also be similar to the damage that is caused by stink bug injury. The first report of pear stony pit in South Africa was in 1965, where they reported that this is potentially a graft transmissible disease, meaning caused by a virus. So, due to the reports of these damaged pear fruits in the Western Cape, Horkro decided to fund a project with the main aim to characterize the association between pear stony pit and the presence of apple stem pitting virus. This project had 4 objectives, which the first one was to survey pear orchards for the presence of pear stony pit disease. So we identified disease-like trees in at least 6 different pear cultivars. We collected samples and then screened all of these samples for the presence of apple stem pitting virus using an RT-PCR assay. What we found is that 71% of the symptomatic trees was indeed positive for the virus. So we concluded that apple stem pitting virus is present in all the orchards that we evaluated, and that there was a positive correlation between the presence of the virus and the disease. But we also identified the virus in asymptomatic trees, but this is not uncommon for virus infections. The second part of this objective was to monitor for the presence of stink bugs to see if they can also contribute to these disease symptoms we observe. We conducted a trial where we deliberately introduced stink bugs onto fruit clusters by using mesh bags, and then at the end of the season we evaluated the clusters for damage. We had a few difficulties with this experiment, and at the end of the season all our test groups had damage. But what we did saw was that less damage was observed on pears that was in bags without insects. So we concluded that stink bugs can contribute to this disease symptom. The second objective was to use high-throughput sequencing to look at the association between the virus and the disease. We subjected 18 plants to high-throughput sequencing, of which we thought 14 were positive for the virus and 4 were negative, based on RT-PCR results. After the data analysis, we confirmed that 16 of these trees were indeed positive for the virus. And we also identified additional viruses, including citrus virus A, apple rubbery wood virus 2, apple chlorotic leaf spot virus, and apple stem grooving virus. The conclusions from this objective was that high-throughput sequencing can provide valuable information that was unknown, and the original ASPV assay was not able to detect all variants of apple stem pitting virus. Then in the third objective, we looked at the genetic variation of the virus. And what we found was that by looking at all the sequence information that we had to our disposal, was that this virus had a wide sequence identity range. And we concluded that there is many ASP variants, and it's therefore important to keep your detection assays up to date to ensure accuracy. In the fourth objective, we developed a sensitive and robust detection assay for all variants of ASPV in South Africa. We rescreened all of the material that we collected during the project for the presence of ASPV, and we found a 78% correlation with symptomatic trees. We then went a little bit further and investigated the symptoms closer, and what we found was that there was at least 2 different categories of symptoms. The first one we defined as large, light brown, stony internal areas that were not present deep within the fruit's flesh. This symptom, or the trees with this symptoms, 56% of them were positive for the virus. The second symptom we described as defined pits on the surface with small internal dark shapes that penetrated deep into the fruit's flesh. And 100% of these trees were positive for the virus. So this project illustrated the contribution high-throughput sequencing can make to ensure virus-free planting material. We delivered a new ASPV detection assay that can ensure clean propagation and planting material for growers. and we identified both the virus and feeding damage as potential contributing factors to pear stony pit disease. To date, the best practice recommendation to prevent disease symptoms is to plant ASPV-free plant material and to eliminate stink bugs from orchards. I would like to thank everyone that contributed to this project.